and also the Nanoletters team for inviting me to present our work. I'm going to talk about DNA origami nanotechnology for dynamic plasmonics. So the field of structured DNA nanotechnology could be traced back to the original idea of Professor Seaman at New York University in 1982. So he had wanted to organize proteins in 3D so that he could study these structures using X-ray crystallography. So nearly after 40 years of research effort, DNA has been widely used as building blocks for nanoconstruction. So then the breakthrough came with the concept called DNA origami, and that was demonstrated first by Paul Rosamond in 2006. The basic idea is to fold DNA into nanoscale shapes and patterns arbitrarily. But the formation of DNA origami is as follows. So it involves the folding of a long single-stranded DNA molecule like a loop called M13 by hundreds of these shorter DNA strands into 2D or 3D arbitrary shapes. So origami is a Japanese word meaning paper folding. Therefore, the folded structure is often called DNA origami. The key to functionality is these capture strands that we can extend from the origami template because they can provide sequence-dependent surface tags for assembly of functionalized entities together, including metallic nanoparticles, quantum dots, dye molecules, among others. Importantly, they also provide addressable bending sites on the nanometer scale accuracy. So this is very important for nanoconstruction because they can provide feature sizes much smaller than those obtained from top-down nanotechniques. For instance, electron beam lithography, focused ion beam etching, etc. So then in this talk, I will be very pleased to show you a number of DNA-based dynamic plasmonic nanosystems with tailored optical functionalities. So the question is, how do we build DNA-based artificial nanosystems with tailored optical functionalities? So if we look into living cells, actually there are many molecular motors which can perform very complex mechanical tasks. For instance, you see on the left, this rotary motor ATP synthase. It can perform the rotating motion, efficiently pumping protons against their electrochemical gradient. There are also linear motors, for instance, kinesin 1, which can walk stepwise along microtubules and deliver proteins as cargoes for intracellular transport. There are also sliding motors. Myosin and kinesin 5 can slide along actin filaments and microtubules, respectively. Then we were inspired by such natural wonders. The first system we have built was a dynamic system with rotation, and that was done by my previous postdoc, Anton Kuzik, who is a professor in Finland. So we deliberately chose to build a plasmonic Carroll system because we know that circular dichoism spectroscopy is very sensitive to three-dimensional conformation changes of Carroll molecules. So in this case, we have this cross-like origami template. It has two origami bundles connected through their hinge area. And then we assemble two gauge nanorods on two bundles respectively, and they are actually functionalized on the surfaces with DNA strands. And then we extend complementary DNA strands from the origami bundles. In the end, due to DNA hybridization, they can be assembled very accurately on the origami template. So we built both the left-handed version as well as the right-handed version. Due to the fact that when light interacts with such a plasmonic system, then plasmons can be excited in these two gauge nanorods. Due to proximity, close proximity of the two gold nanorods, they do couple. So then if we change the angle between these two gold nanorods driven by the DNA-based molecular processes, then we can change the configuration of such a plasmonic system so that we can track the optical signal changes in real time. The molecular processes we have been using are called Toehold-mediated strand displacement reactions. So basically, if we have 3 DNA strands in 3 different colors, so originally this blue strand is partially hybridized with the green strand. Then if I add the red strand into the solution, it can gradually displace the blue strand and fully hybridized with the green strand. So this is the operation schematic. You don't see the gold nanorods. I just want to make the image look simpler. So originally, these 2 locks are open on each, so on both sides. And then for the first Toehold mediated strand displacement, this double-stranded DNA molecule on this arm can be unzipped. And now the 2 blue parts here can be locked through DNA hybridization. Then the system is locked to the left-handed state. If we add return strand through the second Toehold mediated strand displacement, this lock can be opened. Then the system can go back to the relaxed state. Similarly, we can also run the system between the relaxed state and the right-handed state. Just give you some impression about the working principle. You see, we add the DNA driving strands into the solution, and then this DNA lock can be opened or closed, depending on what kind of sequence-specific DNA strands we add into the system. These are the TEM images. On the left, you see the origami frames. And then on the right, you see how gold nanorods are assembled on these origami frames. Then we did optical tracking. Maybe we take a first look at the right figure. So here, the CD spectra in different colors, they correspond to left-handed signal, relaxed state, and then right-handed state. Then we tracked the optical signal at fixed wavelengths around 750 nanometers. Then by adding alternatively the DNA driving strands into the solution, then we can see or identify very clearly 3 different states, namely left-handed state, relaxed state, and right-handed state. So we do see system degradation over time because we needed to always add DNA strands into the solution. Then we figured out to also build a light-driven plasmonic nanosystem because we know light is a very clean energy source. It doesn't produce any waste in the system. So we have this ozobenzene modified DNA lock in red, you see here. So basically, it can be opened or closed in response to UV and visible light alternatively. So in the sense that we can translate light-induced molecular motion of ozobenzene molecules into much larger optical signal changes in a very robust way. So over the last several years, we have tried really different dynamic controls to really operate our plasmonic systems. I have briefly shown you the DNA control, light control. We actually also have implemented pH control, temperature control, aptamer control, among others. So I'm not going into the details of different controls because maybe you have realized until now, I have only demonstrated to you the switching among several angles. You might wonder, so where is the full rotation, like the case of this ATP synthesis machine, which can do the full rotation. So indeed, very recently, my postdoc, Dr. Ling Xin, has realized a plasmonic nanoclock. So in this case, this rotor gold nanorod can rotate against the stator gold nanorod with full rotations, clockwise or counterclockwise. I briefly show you the design schematic. So in the this plot, you see this ring track. So this ring track actually has 16 binding sites in 8 pairs. So basically this bundle, which is used to host this top gold nanorod, can be released from one pair of the so to say the footholds 1 and 1 prime, and then it can be attached to the next available footholds pair, so namely 2 and 2 prime. So in this case, when we add the corresponding driving strands into the solution, then this central scaffold strand can always hold this top bundle when it does the release and attach processes. So every time it steps forward with a rotation angle pi over 8. This is very similar to the case, if you go to an airport, you see such air traffic tower, and this top one is this air traffic antenna. It rotates clockwise or counterclockwise against the tower base. It's very similar to our system. We have these 2 gold nanorods, and then one is rotating clockwise or counterclockwise. Then we did the experimental tracking. And we have 2 colors to identify the counterclockwise rotation and clockwise rotation. So one sees that at different angles, so these 2 sides of data points, they agree with each other very well. It means that the system is fully reversible. So we have also tried another exciting version. So that is autonomous rotation. So that means we don't need to input the DNA driving strands into the solution. So rather, we can let the system rotate autonomously. So in this case, we have the foot, which is a DNA enzyme. So basically along the track, we have RNA substrates. The DNA enzyme can catalyze the cleavage of the RNA substrates in a very controlled fashion. And then in the end, we could also use CD spectroscopy to track such autonomous rotations counterclockwise or clockwise. So I'm not going to elaborate this aspect. So rather, I save time to show you the second example. The second example is a dynamic system with walking. So I have briefly showed you this molecular walker, which can literally perform stepwise walking along microtubules. So it carries this gigantic cargo and delivers it to different target destinations in living cells. We have built a plasmonic version. We call it plasmonic walker. So this walker nanorod can walk along this origami track stepwise. Every time it does a 7 nanometer step forward or backward. So it does the transduction of nanoscale motion into change of optical response in real time. And this work was done by my previous postdoc, Chao Zhou. So in this case, this Walker gold nanorod in yellow, it does the 7 nanometer step forward or backward. So one knows that 7 nanometers is well below the optical diffraction limit. So we were wondering whether we could really use standard optical spectroscopy to track these nano step changes in real time. This is the walking principle. So we add the DNA driving strands into the solution. The foot is released from the original position, and then the new foot is bound to the next station. Then it can perform a 7 nanometer step forward. This is the experimental tracking. The walker goes from the left configuration to a arrow, and then it arrives at the right-handed configuration. And just to give you the impression of the CD spectra for different stations, although the distance difference is only 7 nanometers between neighboring stations, one sees that here the CD spectra actually are very distinct from each other. Then we monitored the CD signal at around 680 nanometers, so we do observe the nano step changes, and we can even identify the walking directions, forward or backward, optically. All right, this is the next example, and that is also the last example I would like to elaborate. That is a dynamic system with sliding. So here, what I'm showing you is this mitosis process. So it is a part of the cell cycle. When replicated, chromosomes are separated into two nuclei. So kinesin 5 is such a plus and directed motor protein, which slide two anti-parallel microtubules apart for segregation of chromosomes along opposite directions. So if one sees the details on the left, so this kinesin 5 protein crosslinks and slide doublet microtubules powered by ATP hydrolysis. Then in our case, we have built an artificial analog. We have two gold nanocrystals which cross-link and slide doublet origami filaments powered by DNA hybridization. This work was done by my previous PhD student, Max Ubang. In order to track the sliding dynamics, we have also positioned a Fred pair on one side of our slider system. So basically it has a donor and an acceptor. And when it does sliding, these two gold nanocrystals can work collectively together to slide the origami filaments back and forth along two opposite directions. I show you also the TEM images. On the left, you see the initial state. So here, this corner shows you the reconstructed image from many of those structures in the background. So it shows this the symmetric configuration before sliding. And then after two sliding steps, it means that it introduces 28 nanometers displacement. And one does see this asymmetric configuration that is formed. Then we did the optical monitoring of the sliding dynamics. In total, we have 5 sliding steps. This is the initial state. There is no displacement, and then it can slide to the left or to the right. We also did the fluorescence intensity tracking as well as the donor tracking. So basically, we can identify very well the sliding steps optically. So we also did the theoretical calculations. It is worth mentioning that, so here in the model, we included the Fred effects as well as the quenching effects. Because here, when we do the sliding, when the slider does the sliding, the distance between the two molecules is changing at certain states. For instance, here, then you see that the donor is also closer and closer to one of the gold nanocrystals, and it introduces the quenching effect. By including both Fred and quenching effects, we found that the agreement between the experiment and the calculation is pretty good. All right, I'm going to summarize. In this talk, I tried to convince you that by the combination of DNA nanotechnology and nano-optics, it is possible to translate nanoscale motion into tailored optical functionalities. So this is very exciting for nanophotonics, as it really opens a pathway to build functional nanophotonic devices with addressability and programmability on the subwavelength pixel level. All right, so in the end, of course, I would like to thank all my students for their hard work and the funding agencies for their financial support. And I would also like to thank you so much for your kind attention. Thank you.